This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 815, recorded on October 8th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and everybody else. I always look out the window to tell you what I think the weather is like, but actually it almost feels summer-like outside. There's a little bit high humidity for this time of the year. There's a gray overcast, patchy clouds. Trees are starting to turn. Starting to think about Thanksgiving already. Crazy.
It's a very nice day today. Very nice day today, yes.
Yeah, it is. Yes.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody.
Hey, Rich.
86 degrees and sunny, headed for 92. Oof. They keep promising us that this is going to let up, but I'm having difficulty managing my expectations. But it looks like maybe a week or so out, they're talking about 70s, but they lie, you know?
They do.
Yeah, there's a profession that they get paid to lie.
Is that monkeys?
Well, except, I don't think it's really lying with intent.
No, I agree.
They're just wrong a lot.
They're just wrong a lot, yeah.
The weatherman can never lie. They're just never right.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's Kathy. 71 degrees. Light drizzle may clear up, and that's the same as 20 Celsius.
We have two guests for you today who are going to tell you a really cool story about COVID, so stay tuned. From Cedars-Sinai Medical Center in Los Angeles, Moshe Arditi. Welcome to TWiV.
Thank you, Vincent. Uncharacteristically cloudy in LA and 66 degrees here.
And from the University of Pittsburgh School of Medicine, Ivet Bahar. Welcome to TWiV.
Thank you very much. It's a pleasure. So it is here also uncharacteristically nice, clear, 76 degree, beautiful day in Pittsburgh.
Boy, we span the coast and in between as well today.
Yeah, we got everything today.
East, west, north, south. How about that? No complaints, folks. We got it all.
Actually, I want to issue a correction before we get too much mail or anybody gets too offended. The weathermen are incredibly accurate most of the time.
Okay.
And I appreciate that.
We just notice when they're not right?
Yeah, exactly.
Because if, scientists are wrong a lot, right? So, but most people don't notice, except us. So our guests today, let me tell you how they came to be. Moshe and I have been emailing for quite a while. We've been complaining to each other about how the variants have been described in various ways, and so then he and Ivet have published this wonderful story that you hear about today. So I said, come on and talk about it. So that's what we're going to do today. But I'd love to first hear your histories, because in the pre-show we were talking about it, and it's just so cool. Tell us, where you're from and where you were trained and how you got to work with each other. Perhaps Moshe, maybe you could start.
Okay, so I'm originally from Istanbul, Turkey, and I went to a French high school there, all boy Catholic high school, and then I went to medical school after that in Istanbul, finished and left in 81. I worked at Yale University for 2 years on surfactant, used in premature rabbits for 2 years, and then went to University of Chicago to do my internship and residency training in pediatrics. Then to Chicago, my favorite town, where I did stayed in Chicago at Northwestern to do an infectious disease fellowship in pediatrics. And then moved to LA. I worked at LA Children's Hospital, and then I moved to Cedars-Sinai, which is affiliated with UCLA, as faculty affiliation, and I've been here 23 years. So I've been working on LPS signaling and toll-like receptor signaling, role of these innate immune molecules in atherosclerosis and vasculitis. And since a lot of people tease me that as a pediatrician, I work in adult inflammatory diseases, I've been also working on Kawasaki disease, vasculitis for the last 20 years. So that's in a nutshell. That's how it is, and we can tell you how we met with Ivet, which maybe I'll let her tell the story.
Sure. Ivet, tell us your story.
Okay, so it is amazingly similar because I was born and raised in Istanbul too, and also went to a French Catholic school for all girls, of course. That was very fashionable then in Istanbul. These were really one of the best high schools. And then continued all my studies in Turkey, actually. I got married, I had 2 sons, and I just did everything, undergraduate, master's, and PhD, everything in Istanbul. And then became a professor there right after my PhD. The university I graduated from, which was the most prestigious Boğaziçi University then, and in the chemical engineering department. So I started as an engineer, and I was a professor there for 15 years until I moved in 2001 to the University of Pittsburgh. And so it's very simple, 15 years in Istanbul, 20 years here. And in my career, I, of course, started as a chemical engineer. I never liked the engineering stuff. I quickly channeled to material science first, polymer physics and chemistry. And polymers, from polymers, I switched to biopolymers, and then gradually computational biology, because in Turkey, the best thing you could do to be more competitive worldwide was to do simulations. We had the best, simulation infrastructure. And then came the year, and I worked every summer for 9 years at NIH, just doing research in the meantime. In the year 2000, turn of the millennium, as you all know, that was the real breakthrough with the human genome. But at the same time, first published by Francis Collins and Craig Venter. And that was the time where bioinformatics, computational biology really became very important and lots of new opportunities. So I joined Pittsburgh to start a new Center for Computational Biology, which soon became a Department of Computational and Systems Biology, which I have been chairing since then. And I also started a joint program with Carnegie Mellon in computational biology. You know how Carnegie Mellon is very strong in computer science and engineering and physics. So I found lots of colleagues there with whom I could collaborate, and found this very strong computational biology PhD program. Now, what I do is mostly modeling and simulations, as we're going to talk about. And I like to use structure. So it is like engineering, but at the molecular level, trying to understand how proteins function, their machinery, and how we can modulate their function, by binding small molecules. I do lots of drug discovery. So, and then, of course, with advances in data, lots of databases in biology, we do lots of data mining, machine learning type of computations. So my lab is essentially, we are tool developers. We simulate everything on the screen, and we collaborate with people like Moshe, who give us stimulating questions, and we try to solve them. So that's where that's how also this research on SARS-CoV-2 spike started.
Okay, Moshe. So you guys you're both educated in Istanbul, and what happened in between?
Oh. Well, I'm sorry, Moshe.
No, go ahead.
So we didn't even know, I didn't even know that Moshe was here in the United States. And a few years ago, all of a sudden he sent me emails asking, are you Ivet Razon, using my maiden name, because that's how. And I would say, yes, I am, but who are you? So I couldn't remember. So that was really, I don't know how many years, more than 4 years for sure, that we had completely lost touch. But we used to play together in the streets. I was the goalkeeper of the team. We used to play soccer and then many other things.
She was the only girl who was on the team.
Great story.
It's lovely. So you connected before COVID, I presume, right?
A few years before COVID, right?
Okay. All right. So I'm not quite sure how to start this story today. There's so many pieces, and maybe you have a better way, but I thought maybe we could start with MISC, multi-inflammatory syndrome C, and telling us, what it is, and then taking your wonderful story from that.
So, and it's exactly how it started. This collaboration. I mentioned that I also have been studying Kawasaki disease for over 20 years. I go to Japan frequently. I've met Dr. Kawasaki almost every year until he died last year at age 90. And it became a passion to me through my training to treat Kawasaki patients in children, and also work with there's a wonderful mouse model of Kawasaki vasculitis, which my lab has been lucky to be funded by NIH for 20 something years for that. So when SARS-CoV-2 came and in March or April 2020, we started to hear about these bizarre cases in children, first in England, and then Bergamo, Italy, and other European countries, about this hyperinflammatory syndrome in kids that follows SARS-CoV-2, that had some findings that are reminiscent of Kawasaki disease, such as conjunctivitis, rash, and oral mucosal changes, which also are present in toxic shock syndrome. And they started to label this as Kawasaki disease-like, and it went further to be called that this is it, SARS-CoV-2 is causing Kawasaki, and then this is Kawasaki disease, So it really bothered me so much that this was wrong, because, since then we learned so much more that these are two different diseases, demographic differences, the older kids are, and then laboratory differences, clinical differences were so much, clear that there were two different diseases. Of course, they may use similar inflammatory pathways that many diseases, trigger. So I became very vocal, in writing or trying to tell that this is a different disease, and if anything, MISC, which is characterized of kids having multi-inflammatory organ system involvement with hyperinflammation that follows the infection 2, 3, 4, 6 weeks later. It's more like it's presenting more like a toxic shock syndrome. Why? Because, it has the components of multi-organ systems, such as acute gastrointestinal findings, abdominal pain, vomiting, diarrhea, myocardial dysfunction, neurologic findings up to 38%, kidney injury, in addition to all those other things, which we don't see with Kawasaki disease, but we do see with toxic shock syndrome. So, and toxic shock syndrome is caused by superantigens. Most people think that only bacteria have superantigens, but then I realized there's a remarkable literature on viral superantigens and the diseases that they've been associated with. So I got in touch with Ivet because I knew she was doing computational studies looking for FDA-approved other drugs to repurpose those drugs for SARS-CoV-2. And I said to her, Ivet, do you think the virus or the spike protein has a motif that may be like a superantigen? and the response was amazing to me. And that was, what is a superantigen? So when Ivet mentioned that, then I will leave it to her now. And I sent her papers about staph enterotoxin B, SEB, or TSST, the typical staph and strep superantigens and diseases that and the structure of those proteins. And a week later, I get a phone call from her, and I'm going to leave her tell the rest.
So at that time, we were already doing some research in SARS-CoV-2 spike glycoprotein. And at that time, I was very intrigued by the fact that, there is between the S1 and S2 subunits of the spike, there is this cleavage site. And there is this insertion that's very unique to SARS-CoV-2. And I was wondering why, what is it so special about it? And I was, at that time, I was trying to see what is unique about this sequence. And I had read a paper by Jean-Pierre Changeux on, the significance of this region, that it resembles some neurotoxins and could be targeted, etc., things like that. So when Moshe contacted me, I said, do you think it might be similar, a superantigenic region that might be there? And of course, I asked what are superantigens and learned that, for example, bacterial toxins are superantigens, so those small molecules that elicit this excessive response, cytotoxic adaptive immunity. And I said, okay, there are some sequences indeed that I've seen in the literature, short sequences similar to bacterial toxins and other toxins, actually, viral toxins as well, viral and superantigens, let's say. And then we started to do a more careful analysis. And maybe I can show you a few slides.
Sure.
Okay. So, for example, one of the things, like background, superantigens, now I learned that they can bind TCRs, but also at the same time, MHC2 molecules. In contrast to regular antigens where you have a peptide, superantigens are themselves proteins. And there is here one of the superantigens that caught our attention, SEB, Staphylococcal enterotoxin B, which binds the beta chain variable domain of TCRs. So when we were modeling the structure and this region, we noticed, when you do a PDB, protein data bank blast, and you find similar sequences, what popped up was actually this staphylococcal enterotoxin B. Let me show you here. So this staph B has indeed a superantigenic region segment that is sequentially very similar to that of SARS-CoV-2, not SARS-CoV, the 2003. But what was even more interesting is that it has what is called a palindromic character. So you can align it, parallel to each other, but also in the anti-parallel way. And then you have even a longer segment that apparently is similar to SEB. Not only the sequence, more strikingly, we also have the structure of that segment, in SEB and in SARS-CoV-2 spike. And you see these are positively charged amino acids located at the same position, you have, lots of similarities in the structure. And then we said this region is definitely sequentially and structurally very similar to a bacterial toxin, SEB. That was the starting point, the discovery of that sequence that closely mimicked a segment of the bacterial toxin, SEB. And then we said, okay, does it bind to the TCR? Of course, it does. So that is located here. And this is how it binds the TCR. And we have lots of, basic residues on the spike and lots of acidic residues on the TCR-V beta chain in particular. So we were very pleased to see that, yes, it does bind also the TCR. Should I continue?
So I have a question. So, well, many, but let's just have one.
Of course.
So this superantigen is relatively short, right? It's sort of like
It is, yeah.
A peptide that would be presented in MHC2, as you say, right? More or less.
The peptide, usually MHC2 presents the peptide alone, right? It is not attached to a protein. So what happens is that in the case of a superantigen, we have a peptide, let's say, again, in this case, about 20 amino acids, 24. But it is also surrounded by other parts of the molecule that may assist also in further binding. So, and that makes, the affinity much higher. And as a result, you have a much larger number of T-cell receptors that are being expressed, or T-cells themselves that are expanded, as you can see it from skewed TCR representations. So here, this is showing as an example, that is the region, superantigenic region here. But there are extra interactions in the vicinity, including adhesion molecule, which serves for intercellular adhesion, which further stabilizes this interaction. So it is the fact that it's part of a protein makes it higher binding affinity.
So would it be interacting as part of the virus particle or on the cell surface of an infected cell?
So it is on the spike, it's part of the spike.
Yeah.
And it is at somewhere in the middle, as I showed here, you see.
Yeah.
So it is, I had a sketch somewhere, the way things interact.
At the beginning.
Here. Okay, here is the host cell. Here is the SARS-CoV-2. This is the spike. This is the spike. And the way it interacts with ACE2, the green line is ACE2. And then there are, I didn't show here, but there are the proteases from the host cell that cleave the spike. So the S2 trimer remains here bound to the virus, and then undergoes a structural change to become the fusion trimer,
Right.
And trigger the fusion process. And then the S1 trimer, after cleavage, is just in this extracellular region. So it may bind directly a T-cell receptor, or the T-cell receptor itself may approach the virus. So there may be multiple ways.
Got it. So you think it has to be released from spike? S1 has to be released in order to be a super antigen, right?
I don't think it is a requirement because it's still exposed.
Okay.
But when it is really, shed into the S1 and S2 trimer, it gets more exposed. So there might be a higher probability of interaction.
So I want a clarification. Does the T-cell receptor engage this superantigen in a fashion similar to other ligands that it engages, or is this a special interaction?
No, it does.
We have a slide on that, Ivet.
Yes, it is very interesting because the way we reported there, the interaction of our superantigenic segment with the T-cell receptor was mostly with the beta chain and with a bunch of acidic residues on the beta chain variable domain of the TCR. And this is exactly actually how SEB itself also interacts with TCRs. And we're going to get there because that's how we also came up with this idea of designing an antibody inspired by what was already known for SEB.
Ivet, maybe we can show the superantigen slide.
Okay, so that is also, you have the T cell and you have the antigen presenting cell. Normally, this is the antigen presented by MHC class 2, but the superantigen, it doesn't even need oftentimes the MHC class 2, but it may also, serve as a glue between them. It has a much higher affinity, binding affinity to the T-cell receptor. Variable beta chain, variable domain of the beta chain.
A lot of people listening probably don't know what a superantigen means, right?
Yeah.
And what's the consequence? So maybe in the context of the bacterial protein, what does it mean and what happens? Yeah.
Superantigen is like an antigen, but it has superactivity. So antigen would be triggering this adaptive immune response, right? TCRs, T-cell will be binding. Now, what happens is that in the case of a superantigen, the response is excessive. So, for example, the number of TCRs that are being expressed or the T cells that are expanded are a few orders of magnitude larger compared to an antigen. And this excessive response, gives rise to a cytokine storm. Moshe can explain even better than me.
Okay.
Yeah, I mean, like Ivet showed in the cartoon, if it's a traditional peptide presented, after an antigen-presenting cell presents the peptide in the groove to the T cell receptor, those are the traditional antigen presentation, and you end up with 0.01 to 0.001 or percent of your T cells that are specific to that antigen. Superantigen bypasses that pathway, attaches from the side to the TCR and MHC class 2, and now suddenly can activate 20, 30, 40% of your T cells suddenly, and with a major release of cytokine. This superantigen term was coined by Pip Americ in 1991, I believe, and initially from the memory mouse
tumor virus. Yeah.
Yeah. Right. And of course, that's what causes flesh-eating bacteria, toxic shock syndrome, and tampon-induced shock. And traditionally has been associated with bacterial, but we know now that there are so many viruses, like I just said, mouse memory tumor virus and rabies virus, HIV GP120 is a superantigen. Ebola has been shown to have a superantigen, and of course EBV, and most interestingly, human endogenous retroviruses have been associated with that been shown to have superantigens, and that's important because we will get into this. Maybe superantigens not only can cause this massive release of cytokines that may send you to shock with cytokine storm, but repeated exposures to it can also cause autoimmunity. And this is what we think what's happening in MISC.
Is Kawasaki's a superantigen disease?
Yeah, so Vincent, that has been debunked. Like it's been like in the 20, 30, 40 years ago, 30 years ago, they found that maybe it's a staph superantigen positive. That those studies have not been, replicated. And although there's a minority of people that think, majority of the people think it's a traditional antigen of virus or viruses that have not been discovered yet. So Ivet, maybe you can take it to do next.
Okay. So now, think about it. We found a segment of the spike, which is sequentially and structurally very similar to this staphylococcal enterotoxin B, which is one of the typical toxins that causes this toxic shock syndrome. All right. And for ACB, there is a huge literature. This has been a system that has been widely studied and also studied by Bettina Vries, actually, at Stony Brook.
Bettina Fry.
I'm sorry?
Bettina Fry.
Bettina Fry, sorry. I don't pronounce correctly. So she has identified, and let me go back to my presentation again to make things easier for you. So she identified 3 antibodies. Let me see where it is. Okay, here it is. So this is from her work, and I can use full screen. So you can see here SEB in the middle, gray, and she identified 3 antibodies, FAB20B1, 63, and 14G8, color-coded here, that bind SEB, that are known to neutralize SEB. And then when we examined more closely, we found that one of them, 63, binds exactly that segment, which is similar to our superantigenic segment on the spike protein.
So for the listeners who might not be watching, those 3 antibodies bound to the SEB in different regions.
Exactly.
It looks like.
Exactly.
And one of them was corresponding
and
to the
then
region
one of
spike.
them binds to that segment, which is similar to the spike glycoprotein superantigenic segment.
Right.
So, because after all, again, as we mentioned, the segment that binds is short, like an antigen, but it is part of the protein.
So these antibodies block the superantigenic properties,
Yes,
right?
that's what Bettina then reported, Bettina Fries, from in this 2014 paper.
Have they been used therapeutically?
So apparently, Pfizer, I was told Pfizer was interested, and then at that time they humanized the monoclonal antibody. Then a CEO change, and the project was dropped.
Okay.
So when we look closer, the way 63, that particular antibody, interacts with CEP, we see that it is interacting with those basic amino acids, lots of lysines here. Lysines that are sequentially homologous to the arginines in this superantigenic region of the spike. And then, of course, we verified, if you wish, that 63, this antibody, also binds our spike. And indeed, we've seen the same amino acid, acidic acid amino acid of the antibody interacting with the basic amino acids of the spike, exactly at this region where the cleavage takes place. So now, binding it doesn't imply a functional impact, of course, but then Moshe's and his collaborators, they also did the experiments to show that there is indeed a decrease in infectivity in a 63, the Fab dose dependent manner. So this has been now published in Structure. And then we started to think even deeper. Should I go on or just wait? Okay. So
I think it would be great just to reiterate this finding. So you have this monoclonal antibody that was known to bind to the SEB molecule, another superantigen, and then you showed that it could block infectivity for SARS-CoV-2.
So SARS-CoV-2, which shares a segment common with SEB.
Right.
So there is, the binding epitope on SARS-CoV-2 glycoprotein is almost identical to that on SEB, not only sequentially, but also structurally.
Right.
So evidently something that binds, one of them would bind the other. So binding was fine, but it was also, we also showed that it reduced infectivity.
Yeah, so this must have been a really exciting finding.
Oh, of course.
Okay.
Of course.
It was particularly exciting because the existing monoclonals, that we use in patients, we were told that you have to use them very early because it only works to block viral reproduction and entry, but it doesn't really work in the later stages of anti-inflammatory stage. This potentially, theoretically, would affect both parts, both blocking viral infection, as well as if the superantigen TCR is shown to be functional and playing a role, it may block that as well.
So what you're thinking is that maybe the inflammatory disease, not MIS, but other inflammatory diseases in COVID might be in a reflection of some superantigenic interaction, right?
Yes, and that, for example, adults that go into, some of them, Akiko's work at Yale, for example, have shown that some patients have prolonged cytokine storm, that goes on, and is it possible that this is playing a role?
Yeah.
SCB is very interesting. Of course, this is all theoretical, and it needs to be proven. But by age, 16, or 60%, 80% of the population have anti-SCB antibodies. But 20% of the population cannot make anti-SCB antibodies, and then you lose your anti-SCB antibodies. These are known from toxic shock syndrome studies. So is it possible that after 70 or 80 years of age, apparently people use their anti-SCB antibodies? Is it perhaps why it's more severe disease at that age? So this is one possibility. The other was very intriguing, is the IVIG has been used in both MIS-C and also in some studies for adult severe COVID, and there are some trials still going on. IVIG is full of anti-SCB. This is why toxic shock syndrome's therapy is IVIG plus steroids. So this is another theoretical thing that we're working on. So that's why this was important.
So some of this discussion gets me wondering about the kinetics of this disease, because MISC, I think you said, can show up pretty late after infection in children. And likewise, the severe inflammatory syndrome shows up pretty late in adults. And I have thought of this as pretty much after the most acute part of the infections are over. So the question arises in my mind, is the superantigen effect sort of a hit and run long-term thing, or do you need to have superantigen present in something like a stoichiometric amount at the time that the disease is induced? What are the kinetics?
All right, so this is the question I get all the time, and so the kinetics is that we're working. These are observations right now and hypotheses that need to be completely understood better. I mean, I feel like we're building a ship while we're sailing, and that's how that's how the whole disease has been, and MIS-C is the same. We found as we go. So, but the way I can envision is that the superantigenic motif, particularly when S1 is shed, and particularly when David Waltz from Brigham has shown that circulating S1 levels directly correlate with disease severity in adults, like who goes to the ICU and stuff. So one aspect is that perhaps these superantigens that are shed in the S1 that are become more exposed are playing a role in cytokine release acutely. That's one part. Then kids with MIS-C, how do you explain that they're coming back 4 weeks, 6 weeks later if this is an acute disease? And their acute disease is just very mild, sometimes even just like a exposure and nothing. But then they come with hyperinflammatory syndrome as an autoimmune kind of like mechanism almost, right? Like kind of like group A strep, and then 4 weeks later you develop rheumatic fever kind of thing, right? And there, I think the hypothesis has to be either a autoimmune interaction that the superantigen is inducing, not only T cells that are going crazy, but also polyclonal B cell activation that eventually turns against itself and may explain all this autoantibodies that us and many other people have reported now in MIS-C. This is one thing. The other thing is that in MIS-C patients, there is another paper that we published with Alessandro Fasano and Lael Yonker at JCI that shows that these kids continue to have virus in their stool up to 4 weeks, 6 weeks, and they have increased gut permeability, and they're leaking perhaps some of these S1s. Indeed, they measured S1 level in the MIS-C patients, and it correlates with disease severity even at 4-6 weeks later because of gut permeability. So while superantigen may cause an acute cytokine release in some patients, adults or others, it may also cause this late onset autoimmune kind of things that we're seeing, perhaps even in long COVID, in addition to MIS-C. But these are all theories that needs to be proven.
So another question that I had was about potential predisposing factors and why it's only a fraction of people who show up with this. I know you've done some work on this, but I'd raised the question. And I noticed in particular a sentence from your PNAS paper saying that speaking of MISC, and this was, I forget exactly when, but no such cases have been reported in China, Japan, or South Korea. That made me wondered whether there was a genetic component or something like that.
Absolutely. And maybe Yvette can show the slide about the HLA finding that, Yvette, if you have that slide, you know. So
I don't think I have it, Moshe. No.
it's the 4 slides I send you, but maybe you didn't include them, but so this is the TCR 11-2 skewing, and then the HLA skewing. But anyway, so what we have published in a JCI paper is that there is obviously a T cell receptor skewing. So you can say, okay, you found this superantigen, and of course the reviewer said the same thing, right? Well, how do you know it's doing anything? You know, I mean, is it really causing any functional? Like, is it there just like for structure, or is it doing? How do you know it's playing a role in the disease, or superantigenic role even? So we had to come in the PNAS paper in the adults that have hyperinflammatory syndrome and show that they do indeed have T cell receptor skewing 24/1 or some other, like couple of them. And that was in that paper. In a subsequent paper in JCI, we showed that in kids with MIS-C, there was specifically T cell V beta 12-2 that is skewed or blooming. You know, so that is a signature in a way of a superantigenic immune signature that specific T cell receptors are skewing. Yale group has shown the same thing, and a French group has shown the same thing. So this is now repeated in 3 cohorts that T cell receptor 11-2 is skewed. In that paper, we start the paper by just saying HLA-associated T cell receptor skewing. So we had, I think, 5 or 8 kids with MIS-C, and among those with severe MIS-C, to our surprise, all of them had a unique trio of HLA finding. You know, I think, so it was A2, B35, and C404, I think it's in the paper. So this 3 alleles, of HLA-1 is somehow helping the superantigenic motif to be presented to the TCR. And this is the first clue, in my opinion, that there is some genetic predisposition. This is why 60-70% of cases are either Hispanic or African-American in this country, and then African origin in Europe. Now, I was very happy to see a MedArchive paper from NIH that just came out 2 days ago, and it's in their abstract as well, that the same 3 alleles are shown in more than half of their MIS-C patients. So this is a clue that why this is such a rare disease. So
would you expect then that this superantigenic region of spike might bind better to that particular TCR? Can you approach this structurally?
Yes.
Actually, that's what we've done also. And we the data we got from our collaborators in Germany was that this subset of TCRs that are observed to be overexpressed in adults with severe COVID-19. And then we checked whether, those TCRs would be binding, and the PNAS paper already contains that. Later on, then a similar type of study for TCR skewing was done with children, the one published in Journal of Clinical Investigation. And that's where, the particular TCR V beta chain 11-2 was found to be distinctly overexpressed. And then we have also shown how the interaction, we have all those results in those papers, how the interactions with the superantigenic region and this particular V beta chain are very strong. Now, there is one more thing, though, I would like to add. A very interesting study caught our attention last year, where it was published in Science by Matthews et al. So what they've done is the following. They have screened 15 MERS, amino acid segments of 15 peptides, let's say, or 15 amino acids on the glycoprotein. So they took each of them and they probed their T-cell reactivity. And they observed this in patients that have not been exposed to SARS-CoV-2. So these are, I have again a slide here, but I can explain. So what happens is that there are many segments actually on the spike that showed a very high T-cell reactivity. So the explanation is that some of those peptides are already shared with previous SARS viruses or common cold viruses. And there might be some people who have gained some kind of immunity, and that might also explain the differences between individuals.
I seem to remember that some of the variants have amino acid changes near the this furin cleavage site. Does that make any difference in terms of being a superantigen? Do you know? Or?
Okay. So Vincent, you just read my mind.
We are going to continue on that topic.
Okay.
So Ivet, there was one slide you showed about the PRR, the polybasic site binding to the TCR. If you get the binding, like structural binding that you showed earlier at the beginning, number 13, I think.
Yeah, that one.
Yeah, and so you see in the middle P681, and all the mutations were just like outside of this, but when the Delta mutant came, P681 becomes, R, right? And so because the motif, this motif that we discovered becomes even more basic, even more basic. So even more basic means that now you can, attach to the acidic furin much more. Therefore, you cleave. You have much more cleave. I think several people, Jason McLean and others that you invited, I think, have talked about this. That the Delta variant may cleave much more. Therefore, two things happen. You make more S2 and more infection and perhaps more virus levels, and perhaps that explains the transmissibility, increased transmissibility. But what people are forgetting is there's also more S1, and more S1 is shed. This is almost like a viral toxin kind of concept, in what Eva Harris, talked about in one of the TWiVs that you had, like NS1 from the flaviviruses that are being released. So we propose that S1 is like and also viral toxin that is being released. And so now here's an observation. So that requires to think that Delta is causing much more severe disease, if that's hypothesis is correct. And I think there are at least 5 papers that are showing that there's more hospitalization, with Delta. I mean, no clear paper shows that there's more severe except hospitalization. But there is a very interesting observation I want to link to MISC.
So, I want to go back to this concept, where actually, at least on my end, everything started. That was, we were focusing on this region of the SARS-CoV-2 where there is a PRRA insert, right, in the segment. And this is followed by the cleavage site, RS. Now, all other family members, actually MERS has also a segment here, but many other SARS-CoV-2 family members, they don't have. So this is an insertion. So that's extremely reactive. You see there are 1, 2, 3 arginines here. In the Delta variant, there is one more arginine coming into play. So such a highly basic region is extremely reactive. So it will But now I think what Moshe also brought up is the reactivity with the antibody might be increasing, but also we have the reactivity with the furin or the protease that does the cleavage at this position, right? So when we were trying to interpret the results, how the antibody is inhibiting the virus or it's the spike glycoprotein from infecting human cells, then we also started to think more closely about, what is happening. We know that we are talking about this cleavage site, just the vicinity, the near neighborhood. And we know that, 63, this antibody similar to 60, which has been discovered for SEB, 6p3 is reducing viral entry. That's our interpretation of the experimental data. But we are talking about the S1, S2 proteolytic cleavage site at the same time. And then we now look more closely what's happening there. So the spike itself, exactly the same region where the TCR and the antibody binds, it also binds this human protease or furin, TMPRSS2 or furin. They both bind at the same position. And they also, exploit those basic residues to have a very tight binding, high affinity. And then if this is the case, there is a competition between the antibody 6D3 and those proteases that all try to bind this insert, PRRA, the vicinity of this insert, right? Yeah. And then when we started to calculate what's the binding affinity. And it turns out that when you calculate the binding affinity between the spike and those proteases versus the spike and the antibody, we found that the antibody binding affinity is comparable to that of furin, one of the proteases. The other one has a weaker binding propensity. So in this image where I have shown both the 63 and TMPRSS2, the protease, bound to the spike. Of course, this never happens because I just superimpose them virtually because they are competing exactly for the same site. So one possible way that we have observed less infectivity in the presence of 63 is that region then was at least partially blocked by 63. And there were fewer furin molecules or proteases binding there. So we came up with this hypothesis that, 63 will effectively compete with proteases thus interfering with the proteolytic cleavage, which is essential for the fusion peptide to be released and for, entering the host cell.
Yeah. So I want to go back 1 minute to the delta mutation, so I can answer Vincent's question. 681 mutation, like P becoming R. So I just said that one of the consequences would be that there will be more cleavage, and then there will be more S1. And when there is more S1, you would expect to have more severe disease. The only thing I can tell you from the MISC as a clinician is that since Delta wave started, we have seen tremendous amount of pediatric cases that shoot up, really. You can go to the CDC MISC website and you can see cases of pediatric cases, and the MISC cases exactly follow each other in the prior surges with the alpha, with the alpha beta, the other variants. But with Delta, we're now seeing severe young kids coming with severe pneumonia to the ICUs and not MIS-C. So you could see that the curve that CDC has on their website shoots up the pediatric cases, but the MIS-C is down. So my hypothesis for that, it fits together with what we're seeing here as a clinician. We don't see much MISC anymore. We keep seeing acute severe disease, which we didn't see before. What was the history of MISC? Very mild disease initially, and then 6 weeks later, 4 weeks later, severe MISC. Now, very severe disease at the beginning and no MISC. In my opinion, that's because of perhaps the 681 is doing something, that mutation, and pushing the more shed spike that is causing more initial severe disease. And when you push so much Th1 and so much severe disease, perhaps the body turns off the autoimmune part for later because you're just like dealing with the acute disease initially more. But this is a very interesting observation.
The opposite of what you would predict, right?
Opposite what you would have predicted.
Yeah. I assume there are no animal models of MISC that you could test all of this in, right?
No. But we're trying to give spike to and then see what happens with repeated spike to see if there's an autoimmune thing. Yeah.
So the question that was on my mind the whole time I was reading this stuff is, what about vaccination? Why doesn't vaccination trigger a superantigen response? Or, maybe there is something about vaccination that's
this
related.
Is something I think I wrote to Vincent one day. I think David Walt with Alana Ogata published another paper where they measured circulating S1 after vaccination. And they have found that, in the first 8 days, there's a small amount, 50 picogram, 70, 80 picogram, of circulating S1 after vaccination. For the first vaccination. In the second vaccination, they couldn't because maybe antibodies, interfere with their ELISA. That may play a role in, maybe the headache and the side effects or whatnot. But it's not enough or sustain. And you probably need the rest of full viral disease to really cause this, and not just like one spike or for a few days or something. And that's the only explanation I have.
So one other thing is, if you look at the literature, all the structures, cryo-EM structures that have been resolved for the spike, they have replaced this PRRA segment, mutated it, because this is a highly reactive and a flexible region that doesn't lend itself to structural stabilization. So I am not sure, but we may want to check whether in the vaccine, the sequence that is being used, did they keep the PRRA segment, which is, highly superantigenic or not? I think we need to check. That's a very good question, though.
I think I know this, because I talked to Jason McLellan about this, and I think the PRRA motif was changed, mutated.
So then there won't be
in the Johnson and Johnson vaccine and the Novavax vaccine, not the other ones.
Oh, interesting. Interesting. Yeah.
But the mRNA vaccines have a C-terminal 2 prolines added, right? What does that interfere with the site, the furin site at all, or is it distant from it?
C-terminal will be at the S2 subunit.
It's too far. Okay.
So it is at the other.
C2. Okay. Got it. S2. Got it.
Yeah.
But before we, I don't know when you want to wrap this up. I just want Ivet to show us, in my mind, what is the most interesting observation she made, which are the neurotoxins.
But I think we ran out of time.
Well, you are welcome to stay on. I just was being respectful of your time.
Okay.
But,
so sure, I can show very quickly. When we did the analysis, because we didn't know where to look at in the very beginning, so we scanned all the sequence, and that was a study from the lab of Jun Wu and others in the year 2004, that's published in Viral Immunology, after SARS, the first SARS pandemic. And they looked at whether there were any superantigenic, toxin-like, or adhesive molecule-like segments in SARS-CoV-1, the first one. And then they did identify then a bunch of motifs, including a few neurotoxin motifs, here highlighted in green. Now, we checked whether those motifs were conserved in SARS-CoV-2. And then we found that one of them actually is very, highly conserved and quite long, actually. It's not a short segment, it's quite a significant portion of between residues 299, threonine 299, and tyrosine 351 of the spike. And the end of it, one end of it here, the C-terminal end, is very exposed, like the receptor binding domain or the N-terminal domain of the S2 subunit or entire spike. So we were very intrigued by this neurotoxin motif as well, which might be relevant to these effects, we observe in the central nervous system, especially in the long COVID case. And then we wanted to see whether the TCRs that have been identified to be overexpressed, expanded in patients with a severe COVID, and in this case, we are talking about adults, whether those TCRs could also bind this neurotoxin motif. And it turns out that they bind this region even stronger than the superantigenic region. So that is now another motif, neurotoxin-like motif shown in green here, where the TCRs will be binding as well. And this is certainly, in progress. But I just, mentioned the science paper of Matthews et al., where they have identified in people who had not been exposed to SARS-CoV-2 some cross-reactive epitopes from the spike. And it turns out that that particular neurotoxin-like motif shown here turns out to be one of the most highly reactive segments on the glycoprotein. So we are right now pursuing this line of research to better understand the role of this particular segment, which is actually partly conserved between the two SARS, and has been verified here to be cross-reactive, even in patients who have not been exposed. So I think maybe Moshe, if you would like to add anything else, but I think
what we're doing is that
we are very excited about this motif now.
We now have these neurotoxin motif that Ivet just showed, and we made a recombinant protein, and doing doing experiments with blood-brain barrier opening and going through. I mean, people have shown that S1 spike protein opens up the blood-brain barrier. And so it's not inconceivable that these shed or neurotoxins can pass through the blood-brain barrier and may lead to some of the neurologic findings that we see in acute COVID neurologic findings. I think that's really interesting. I just want to finish with one thing that, when I started to look into this and the role of superantigen, I came across a paper in 1982. I love these old papers. So David Eschenbach from Seattle, who was an obstetrician, who wrote this incredible paper. And you could take out toxic shock syndrome in the title, and you can put long COVID in there. And the title of the paper is Persistent Neuropsychological Sequela of Toxic Shock Syndrome, 1982. And he describes, 16 or 10 or 12 women who go into toxic shock syndrome, and then a year later they are describing syndromes of like neuropsychiatric of headaches, not concentration enough, or cannot really ability to concentrate. It's almost like you're reading the long COVID, from today, and I thought it was very interesting. To me.
Well, I wouldn't call that very old. I'd call 1939 an old paper.
All right, sorry.
1982 is when I started at Columbia.
It depends on your perspective.
Yes. So
what you're describing sounds like the brain fog that is the term that people are using now.
Exactly.
For the long COVID. Yeah.
Yes.
Do you have any sense for what the peptide might be hitting in the CNS to do this, or is that something you're interested in finding out?
That's too early to do
Okay.
that kind of stuff, but.
Now, I have one last question, which is this amino acid sequence must be an accident that it has some other function. It needs to be cleaved by furins, right, to improve infectivity. So it's an accident that it causes the T-cell receptor amplification and so forth, right? Because that would be of no benefit, evolutionarily speaking, for viral selection, right?
Well, superantigens are generally thought as virulence factors to help the microbes to do the disease.
Well, but
For example,
that's true, yeah. But
rabies virus superantigen is used apparently to bring the virus from where the virus is in the muscle all the way to the nerve endings. It plays a role by activating specific T-cell receptors. So I have no idea why the origin of this might be, or what the role of it is.
I just, it doesn't, to me, this is my particular view, it doesn't make sense. It doesn't seem like it would be selected to cause this MISC, because it's late and it has no impact on transmission, which is a major selection for. So I would look at it as an accident, especially since SARS-1 doesn't have it, but who knows? Anything else you wanted to mention? It's a really cool study. Oh, is there any implication for treatment? So should we look at everyone's HLA-1 and say, okay, you are at high risk for MISC and be ready to treat them? Would that be one approach?
Well, one thing I read was in COVID-19 adults, there were already an HLA type study, like who would get sicker. But for kids, this needs to be, repeated and in other cohorts. I'm aware of like 2 other cohorts that are finding similar HLA finding. But once we confirm this, that may give us a clue, which patient may develop more severe, HLA, disease,
It seems to me that the antibody would be a good treatment, right?
Yeah. So
almost all of the antibodies right now, they target the RBD, the receptor binding domain.
Yeah.
And, whenever there is a mutation there, you may have an effect, evasion risk for multiple antibodies. Now, 63 on the other hand, and there is one more antibody actually called 4A8, they bind another region, and they have now shown a reduction in infectivity. So I think using this antibody in cocktails might together with RBD binding antibodies might be a good strategy.
So of course, to try this, you need to titrate in appropriate animal models. If you use an animal model where you give lethal virus and then you give this antibody, you may not necessarily see a suddenly reversal of death.
Yeah.
But to look at the pathology or level of pathology in the lung might be the way to go, perhaps, to try this. And we're trying to work to have collaborators to help us those experiments.
Fascinating story. Really nice example of how you can start to figure out pathogenesis, right?
What I like is
mechanisms.
It starts with an idea.
Yeah.
That's just an idea that I probably would have discarded and gone to lunch, and then experiment after experiment and paper after paper using different approaches turns out to support the idea. Okay, so it's a great story. It's a great journey.
Right.
Still in progress.
And we're still continuing this
that's right.
journey.
All right. Moshe Arditi from Los Angeles, thank you so much for joining us.
Thank you, Vincent.
You're welcome.
And
You're
Yvette
welcome.
Bahar, University of Pittsburgh. Thank you both for joining. Really appreciate it. And good luck. Keep it up.
Thank you.
Bye-bye.
Thank you for inviting us.
Thanks. Good
Pleasure.
talking to you.
Pleasure.
Great. Good story.
I think it's cool. I don't know of any other pathogenesis where they're starting to sort it out, right? MISC looks like they have a mechanism, right? That's good.
So I was reminded of their story when they started to refer back to the streptococcus and the disease is an autoimmune disease that has a lookalike molecule someplace. I was at Rockefeller when that data was first discovered by Zabriskie and Freimer. And I remember everybody getting very excited about that because rheumatic heart fever could be now explained by waiting to treat. And then all of a sudden you get another disease, which is even worse than the scarlet fever, the strep throat that you had to begin with. So nothing is unrelated.
Staphylococcal enterotoxin B, SEB. She said that fast a lot. You might have missed it. So
exactly.
SEB. And that's the superantigen, and which triggers TSS, right? Toxic shock syndrome.
Yeah, exactly.
And the other thing that maybe people got lost with, so the T cell receptor normally would recognize a peptide presented to it by MHC, right? And so this superantigen is really good at recognizing the T cell and somehow amplifying certain ones so that they're way higher than they should be.
Basically, the way I see it is it activates those T cells.
Right.
Okay, which means that they divide and proliferate, and they also send off all kinds of cytokines.
That's right. And
that's when they talk about skewing, that's what that means is you've amplified a certain population of T cells that this superantigen reacts with, which is another terrific thing because they basically predicted in their model that something like that should happen. And then they go look at the kids, and sure enough, they have a whopping fraction of their T cells, the kids with MISC, whopping fraction of their T cells are of a certain type that react with this superantigen. Amazing.
I don't know if it's actually, where's S1, right? I guess it's being shed at entry, so it's got to be coming from virus particles attaching to cells at
I would assume
cleavage, and
so,
the S1 is shedding off, right? Because there are a lot of questions on the biology that need to be addressed. But in one of their papers, they said you can't change the sequence because then you have no less infectivity if you change the furin site, and then you can't dissect the two properties. I wonder if you put it into another virus. Oh, that's gain of function. Sorry. But you could put it into and if you had an animal model for another virus, say an animal virus that doesn't infect people, put it in and see if it triggers MIS or some inflammatory syndrome that you wouldn't see otherwise, right?
So, Vincent, I noticed that they didn't bring up the issue of blood type. I was going to even raise that, but is that an old story now that's not being adhered to anymore, or are there certain blood types that are more susceptible?
My understanding is that was no longer held up on further examination, the connection with blood type and severity. Yeah.
Okay.
But what is the frequency of this? I wanted to ask them. Does anyone know the MISC?
Kawasaki?
No, not Kawasaki. MISC, like what?
I don't know. We should have asked him that. It's not very common, I think.
No.
But I just love that you may have a mechanism to explain it, right? Immunological, molecular. And isn't it great? You look at this and you see it looks like a superantigen. Wow, that must have been cool that day, right? Anyway, it's a great story. That's why I wanted them to tell it.
Right.
Let's do a couple of emails. Oh, you have a public service announcement, Kathy.
Yeah, it's something that I only just found out about, but we lost an important adenovirologist, Bill Wold. He died in June, and I only just found out about it last night. So Bill Wold has he spent his entire scientific career at St. Louis University, having been trained in Manitoba, so he was Canadian. And he was about to retire in August and
Oh,
moved
dear.
to Vancouver Island in Canada.
Oh, brother.
But so what I want to tell you about him, we have two links. One is the obituary from the web, and also a Journal of Virology article that a number of people who were with him at St. Louis University wrote together. So it's just a longer, really nice tribute about his life. So he was an adenovirologist, and he was E3, basically. If you wanted to know anything about E3 and human adenoviruses, Bill Wold was your guy. And one of the things said that he was really generous with, as a citizen, he generated over 250 viral mutants and all kinds of antisera and monoclonal antibodies, and was very happy to provide them to anybody. And I was the recipient of that. Back on some of the kinds of things that he did, E3 is conserved among all the adenoviruses. I mean, it's always there, and yet it can be quite different from one adenovirus to the next. And the early things that the rap about it was that it was dispensable. Well, it was dispensable for growth in cell culture. And Bill argued that, no, it must be important for something in the virus, in pathogenesis, in the natural host. And so he really worked to characterize that. There are a lot of nice tributes, both in this Journal of Virology article and online. He was known not only for his scientific achievements and leadership at St. Louis University, where he rose through the ranks and became the chair of the department. He was a supportive mentor, caring person, outstanding teacher, and a dear friend to many of us. He was a favorite golf buddy, a passionate running coach. He ran marathons, and this is something I didn't even know about him until now. So he will be missed by a lot of people and had a really positive impact on the field.
Remember, Harry Ginsberg used to say that too. It's dispensable for cells, but must be doing something.
Yep. E3, really important. And yeah, so too bad.
It's one of the genes they take out in vectors, right, to give more space.
Right. And that, and so there were two ways that Bill sort of addressed that. And one was, let's figure out what all these E3 genes are doing. And the other was, if we can take it out, then maybe we can use it as an oncolytic vector. And so he had a lot of connections with working on cancer and things like that. So, yeah.
It's a day when you could focus on one gene.
Yeah. Well, one region.
One region, your whole
E3
career.
has a bunch of
How many genes
Yep.
are multiple genes?
Oh, yeah, 6 or 8 for some of the human adenoviruses.
But still,
yeah.
to focus on one region your whole career, people did that all the time. I don't know if you could do it anymore.
Yeah.
And publish papers with 2 or 3, 4 authors.
Yeah.
Oh, and his people were all very loyal to him. So the postdoc who left my lab was with him from 1996 till 2020, when I think he saw the writing on the wall that Bill was going to retire, and he changed labs. But all those other co-authors on the JV article, I think many were with him for years and years. Yeah.
Yeah, I met him a few times. All right, Rich, can you take the first email there?
Sure. Hal writes, first of all, thank you for your excellence. I am a 64-year-old, relatively healthy and fit internal medicine physician practicing in southern Maine. My second vaccination was on January 20th. Recently, just this week, I contracted SARS-CoV-2. This is day 5 and nothing more than an annoying cough, mild fever, and a runny nose for a few days. I'm almost back to, in quotes, normal. Yay, vaccines work. My question is, do I truly need a booster? Fully vaccinated plus COVID, more than likely with Delta variant, in that order. I know there's no negative outcome in getting the booster, but is there truly a benefit in this scenario? If so, what is it, and when would be the best time to get the booster shot? Well, I'll give you my spin on this, and others can weigh in. I would say, first of all, and most important, I don't know. Okay. And I don't think anybody knows. We do know that there are studies that say that if you've had the disease and then get vaccinated, you have a superpower, okay, that you are immunologically cooler than either infection or vaccination alone, okay? And I would presume that the reverse is also true, that if you are vaccinated and then get sick, that you have a superpower. But there are no data that I am aware of, so I don't know. Your thinking is all right. Sounds to me, if I had to guess, I would say that at this point, a booster shot, although no harm done, is superfluous.
So a superpower, that's different from a superantigen, right?
Yes.
We got the super episode today. I would agree with that. But I like that Hal has thought about this, and also, he says, I contracted SARS-CoV-2 virus. Good for you. And yay, vaccines work. They do.
Yeah. And this is what's going to happen over time, over the years, as we have said many times before. My belief, my prediction is that the virus is not going away. And, depending on what happens to the vaccination programs, I would kind of expect that all of us, if we live long enough, are going to, even having been vaccinated, going to get boosted by the virus itself. Okay. So there you go.
Dixon, can you take the next one?
Oh, you gave me the long one. Sure.
No, it's
absolutely.
Lamont. It's a short one.
Lamont writes, what I find interesting, there is a documented transmission change of breakthrough infections in fully Pfizer vaccinated individuals. What I sort of expect is this will be abused to continue to argue that the vaccines don't work with Delta. And that's a follow-up to the comment that was just made.
So this is an emerging infectious disease, very short. It's called a research letter. Breakthrough infections of E484K harboring SARS-CoV-2 Delta in Lombardy, Italy, which was the region really hammered initially, if you remember, at the beginning.
Yes, that's right. That's right.
And the last TWiV that I released, one of my guests worked there and studied it firsthand. So this is an interesting case. It is someone who got infected, and then his mother, and they're all vaccinated, and then the mother who's vaccinated got infected, and then somebody else close to them, and the genome sequencing suggests that it's all the same virus that they transmitted to each other. So they're thinking these vaccinated people were transmitting it to each other. Yeah, I mean, it's a couple of people, I agree, and I don't know what the bigger picture is. But of course, yeah, a small study like that will be used to say the vaccines don't work, for sure.
But no one died.
Oh,
no one died.
Nobody died. That's right. I think
it worked.
Pretty fatigue, headache, myalgia, dyspnea, fatigue, none required hospital admission. Yay!
So a cold ran through the family.
It did.
Yep.
Yay! Vaccines work.
That's it.
Yeah.
To quote
another proof.
Hal. Kathy, can you take the next one?
Yes. Jeff writes, "Hi. I was trying to explain the immune system to my 12-year-old son in simple terms. I don't think that there are really any simple terms for the immune system, which is what makes it so fascinating. But anyway, I came up with what I think is a pretty interesting metaphor of musical recordings and their being played. The idea is that there is a memory, the stored music, which has no specific playing volume, but a spectral analysis would reveal many combined frequencies, each with a relative amplitude. When played, there's an overall amplification that can be set regardless of the recording's inscribed amplitude. This playing amplitude expresses all of the memories at their relative amplitudes. Then we talk about immunomodulation. It is the external overall amplitude that we're talking about. And the T and B memory is the spectral decomposition. The T and B memory inscription process is similar to our recording of music. The system records on the record a response to what is going on in its environment. Although we don't usually represent music in terms of its spectral decomposition, there's not reason that we couldn't. And if we did, then the TB immune memory system would actually work similarly to that way of recording music. Anyway, I'm not sure how far this metaphor or analogy could go, but I thought it was fun at the time. What I wanted to ask you, though, after all this, is what is the best book on the immune system for a 12-year-old for under $150? So, for instance, Janeway is out, although I could probably get a previous edition cheap. Cheers, Jeff. So, if your 12-year-old could understand this analogy with spectral analysis, combined frequencies, amplification, and so forth, then kudos to him and to you for knowing that he could understand that. Because I think it's a pretty complicated analogy.
It is.
But anyway, maybe it works, and maybe it works for some people who think more about music in physics terms, which reminds me I have the chance to go to another Saturday morning physics about music in the near future.
Nice.
So I'll have to do that. The book that we've suggested in the past that I have and I refer to is about a 1/4 inch thick, 8.5 by 11 format paperback by Lauren Somperak called How the Immune System Works. And you want to get the 6th edition because immunology is always changing and you want to have the most current one. So How the Immune System Works.
Nice.
Is my recommendation.
So Susanna Lopez, she's not written anything that would do the trick. Has she yet? She needs to tackle this one.
I think she might have said she's working on it.
Yeah, she's written some things that kind of get at some aspects of the immune system, and then I don't know. It seems to me that her books are targeted more
Younger.
Younger age group. But that could also work for understanding immunology.
It's true.
So,
The other answer is as soon as we understand the immune system, that book will be written, but it hasn't been done yet.
Right. All right. This next one is from Johannes. Hi, Twivsters. I'm a little behind in listening, so I am sure someone already told you. But just in case, in TWiV, rats, more coronaviruses from August 29th. You were talking about a ligation method that leaves no traces and that you didn't know of one. Gibson assembly would be my obvious example, as I use that in the lab all the time. All you need for that is fragments that have overlapping sequences of 20 to 30 base pairs in an enzyme mix containing a 5' exonuclease that generates long overhangs, a polymerase that fills in the gaps of the annealed single-strand regions, and a DNA ligase that seals the nicks of the annealed and filled-in gaps. And voila, no traces of ligation, because you do not have to use restriction sites if you synthesize or PCR amplify your fragments, and no recombination scars like in gateway cloning. Yeah, I forgot about Gibson.
Yep.
Have a great day from Perth, Australia. Okay. Let's do one more round. Rich?
Adam writes, greetings, Vincent and everyone. I love TWiV, but I got to take a very slight exception to your discussion of Star Trek red shirts in TWiV 802, which was in every other respect a terrific episode. God, a Star Trek boo-boo. This is terrible. How many times has it been said on TWiV when discussing statistics, check the denominator? If we apply that principle to the Star Trek red shirt phenomenon, we get a different story than the usual narrative. By the way, the usual narrative is that if you show up in a Star Trek episode wearing a red shirt, you're doomed. Those are the characters that wind up getting killed. While there's actually a book written about this, or a novel about red shirts. While it is true that more red shirts, engineering, communications, and security personnel, that's their uniform is red, died in the 3 seasons of Star Trek than either gold shirts, command positions, or blue shirts, science and medical staff. If we look at the population of the Enterprise, we see something else. By the way, I've heard rumors that there were some Star Trek shows after the original series, but I suspect that may be fake news. Anyway, by raw numbers, the deaths break down as red shirt, 25, that is 58% of total deaths, gold shirt, 10, 23% of total deaths, blue, 8, 19% of total deaths. The Star Trek Enterprise Technical Manual Guide states that there were 239 red shirts on the Enterprise, 55 gold shirts, and 136 blue shirts. This means that the death rate by shirt color is red shirt, roughly 10%, gold shirt, roughly 18%, and blue shirt, roughly 6%. That means the safest position statistically is to be a blue shirt science officer. Next is to be a red shirt engineer security person, and the riskiest position or highest death rate is with the gold shirt command officers. Credit for this calculation is not mine, but goes to mathematician James Grimes, whom some of you may know from Number File or Singing Banana. It's a new one to me. I got to look that up. Keep up the great work. I love TWiV and all the microbe TV offerings. Thanks for all the knowledge and entertainment. Sincerely, Adam. Well, we stand corrected. And I love the data. It's marvelous.
That's it. That's what does it. So I don't know anything about this, but I was on some Kansas Medical Center show, and one of the doctors said this, and so I just passed it on here on TWiV, and I think you guys may have agreed or something.
Well, there was a. I think one of the reasons this came up was that there was a photographic meme going around for a while.
That's right.
With 4 Star Trek crew, 3 of whom were wearing masks and 1 of whom wasn't, and the one who wasn't was a red shirt.
Right. Right. Okay. All right, Dixon, now you get a longer one.
Now I get a longer one. Georgia writes, "Hi, Vincent and crew. Listening to Shane Crody's responses to your questions and impact of timing of boosters, I wondered if I might apply that to my situation with the shingles vaccine. I got the first Shingrix shot at the end of August. The timing for the second shot is identified as 2 to 6 months. Based on the discussion in the above episode, do you think it might be better to get the second Shingrix injection towards the later time frame? Is it fair to extrapolate response to COVID-19 vaccinations to the Shingrix vaccine as far as timing of the booster is concerned? Some background. I developed shingles of the trigeminal ganglia on the maxillary branch V2 early in July after I finished a 10-day course of acyclovir, 800 milligrams per 5 times a day. The shingles resurged and expanded to include the ophthalmic branch, V1, and I took another course of acyclovir, and my eye doctor put me on a 90-day course of 1,000 milligrams of valacyclovir as soon as I finished the acyclovir. I read up on at the CDC on Shingrix. How come I can't say that word? Shingrix, and confirmed that the use of antivirals wouldn't interfere with getting the vaccination. Alas, I was left with some nerve damage, but I haven't given up on the hope that it might eventually improve. Love all the microbe TV shows and listen faithfully, Georgia. I'll practice that word over the week. Sorry.
If they say 2 to 6 months, then you can go anywhere in that range.
That's a pretty broad range, though, isn't it?
Yeah, and I doubt if there's any data that says that any particular interval is better than any other. I wouldn't necessarily translate experience with one vaccine to another. Okay, so whatever. I would, in your circumstance, I might want to get boosted as soon as possible.
Yeah.
It sounds like I'm sorry about your situation. That sounds like a pretty awful experience.
That's right.
For a while, the Shingrix, there was a shortage of it. And so I was faced with, I had gotten the first dose And I was toward the end of the 6 months, or maybe just outside of that, before I was able to procure a second dose. But now I think that's long since eased up. So
Yeah, I think I got my second dose at 7 or 8 months just because I forgot. At any rate,
Did you guys
a little behind the scenes here. There's a good. I love this sentence that read up on CDC on Shingrix and confirmed that the use of antivirals wouldn't interfere with getting a vaccination. And the deal is that the Shingrix vaccination is a protein subunit vaccine, so there's no virus involved.
Yep.
So an antiviral is not going to deal with that.
Did you guys have a strong reaction to Shingrix? No.
Little stiffness in the arm, not bad.
No. Okay. Kathy, you're next.
Yes, Christos writes, "Hello, I love your show. Keep communicating science. You are very unique in what you're doing. I want to hear your opinion on the use of TCID50 to determine virus titers, either with in vitro assays or in patients. I keep seeing it on SARS-2 papers, and to be honest, I hate it. I feel like quantification of infectious particles should be measured only in plaque-forming units. TCID50 mixes up CPE, general pathogenic effects, syncytia, and so forth. It doesn't seem right for viruses like SARS-2. Am I wrong? Here's a neat little paper describing 2 mink outbreaks. Cheers, Christos." Okay, so I will tell you my take on plaque assays versus TCID50s. They're both using cytopathic effect, and in some cases, maybe syncytia or whatever, to measure a plaque-forming unit or some kind of pathogenesis that allows you to score particular well in a TCID50 assay. They're both quantitative, and if you just state your units consistently in TCID50 units or plaque-forming units, I think it's fine. One of my colleagues here doesn't like plaque assays and says that TCID50s are better. It may depend on your virus, what you're used to, and so forth. But really, in my mind, they're measuring the same things. But now let's hear what Vincent and Rich think.
Well, I don't do TCID50s, but not all viruses will plaque. So sometimes you have to do a TCID50 if you want infectivity. And I noticed that companies tend to use TCID50s a lot because I think you can do more high throughput with TCID50 than plaque assays. It's hard to do in a high throughput. But I agree with it. They're both measuring CPE. And you could even do both and make an equivalence of the two if you wanted for your particular virus.
I agree. I have done only plaque assays, except for I had a student once. We were studying a pox virus of seals, and actually isolated this thing, and it wouldn't plaque, but it would cause a cytopathic effect. I found it. To me, it's always been cumbersome because I'm familiar with the TCID50 as you got to visually inspect all these wells and decide where the cytopathic effect is. But I think there are sort of staining methods that you can use where you can use a plate reader. And in that case, it would be a piece of cake. And I think some people use TCID50 because it's easier.
Yeah.
If you can do that kind of thing. So, but I agree. I think they're both measuring virus replication. So that's the key.
And, as Kathy said, some people like one, some people like the other. It's fine.
There's something satisfying about a plaque because you can sort of relate to the notion that a virus did this. A single virus set this off. But that doesn't really tell you everything you need to know about the population of viruses that you stuck on the plate, okay? Because plaque assays, are themselves not perfect in quantifying all the potentially infectious particles in the solution. So from that point of view, they both are a kind of a relative measure of what's going on, but they're both measuring infectivity. So cool.
This paper is very nice that was sent to us here by Christos about mink outbreak, SARS-CoV-2 outbreaks on mink farms in Germany, Greece. And it's very detailed. It's cool. tracks it really well. By the way, they killed all the farmed minks in Denmark and Netherlands after the outbreak. I also learned that on our latest TWiV. Last one is from Bev. You'll probably have lots of people telling you this, but just in case, dogs' eyes refresh at a faster rate than ours do. So what we see as a moving screen is a flickering mess. Here's the first link I found on it. So we were thinking about whether dogs see TV. And this article in the Nest is, do dogs see the TV the same as humans? Dogs see a whole new picture when it comes to TV because of the flicker rate. Yeah. So what do they end up seeing? They don't have the same visual acuity. With the advent of high-resolution broadcasting, hopefully television producers have tapped into the canine market. Dog TV, a channel devoted to programming for dogs, launched in 2012. It broadcasts a mix of relaxing and stimulating content designed to reduce stress, boredom, and loneliness while dogs are alone in the house. My neighbor, who has like a dozen schnauzers, they rescue them. Their TV, when I go to my car, I can see the TV and it's on 24/7 to entertain the dogs. You know, you could go out any time of day and it's on. So they see a flickering mess, I see. But clearly some dogs are mesmerized by TV.
Yep.
Well, flickering mess could probably be pretty entertaining.
Could be. All right, it's time for some picks. And I am really looking forward to hearing the next installment of Dixon's Top 10 Comedians.
Well, this is number 4, as I recall. And he should have been number 1, but I didn't alphabetize them or anything. I just randomly picked them. Mel Brooks is by far the most versatile of all the comedians that I've become attached to, and it tickles my funny bone. Every time this man says something, it's usually funny, and he intends it to be, and he's quick, clever, a remarkable individual. He's made movies. He's done stand-up. He's selected incredible casts for some of his movies. And this is a compilation of his life, basically. It's a pretty long YouTube documentary on Mel Brooks. And he starts out by dancing with his wife and singing Sweet Georgia Brown in Polish. And the documentary ends with the same stand-up, except that his wife is now sitting in the audience and he has to go get her. She doesn't want to do it. And he drags her onto the stage. And then the moment they get started, she doesn't miss a line. She does all the steps correct. It was just incredible. They were a wonderful couple. There's some very touching and warm scenes from Gene Wilder as he recollects all of his experiences with Mel Brooks. And Carl Reiner, of course, figures prominently in this as well. One of the best lines I thought I've ever heard from him was when he was being interviewed by Carl Reiner as the 2,000-year-old man. And of course, they sent him first to the Mayo Clinic to make sure he was 2,000 years old. And he says, well, I'm not quite 2,000 years old. He says, in September 16th, I'll be 2,000 years, but I'm not quite there yet. So no, let's not push it. You know, he was even funny at that level. So Carl Reiner asked him once, he said, you know, it must have been rough being 2,000 years old. When you first started your life, there were no medicines, there were no doctors. He said, what did you do for illnesses? He said, what do you mean? What kind of illness have you got in mind? He says, well, what if you got diarrhea? What would happen? He says, well, you took clean peaches. That was instantaneous, out of nowhere. The man was, and still is, the fastest thinking, funny man I've ever heard. So that's my pick.
I'll have to have a look at this. It looks good.
Yeah.
Oh, you'll like it. You'll like it.
You know, another quick one was Joan Rivers. She was really good. I don't know.
She was excellent.
I don't know if she's on your list, but she was really rapid. She could have conversations.
She was. You're absolutely right.
In fact, someone put an old clip of her on the Johnny. So Johnny Carson apparently got her started.
Yeah, but there was a disconnect there.
Yeah, well, then when
afterwards.
when he was retiring, she wanted him to give her the show, and he didn't, so she
That's
got
right.
mad at him.
That's right.
But
Reneged.
I watched this clip of her where she came 20 years after her first appearance, and she
Yeah.
was so quick and
Yep.
saying funny things. It was amazing.
Yep. Totally agree.
Kathy, what do you have? Sorry.
Born Melvin Kaminsky. He's 95 years old.
Yeah.
Right.
He was a corporal in the U.S. Army, served in the Battle of the Bulge.
Exactly.
Melvin Kaminsky. Kathy, what do you have for us?
So I have some watercolors that are a virtual exhibit, and I want to read you something written by the artist. I'm a painter-photographer who spent 12 years at the Art Students League in New York studying with Mario Cooper and Dale Myers. I am a member of the Salmagundi Club in New York City. These are representative of some of my recent watercolor paintings that I entitled Germination. I live in Fort Lee, New Jersey with my wife, Marlene Bloom, an accomplished artist who paints in oil on canvas. To see my photographs, please go to despommier.photoart.com. So what you have now figured out is that these beautiful watercolors are by Dixon. And I didn't even know that he had this talent or this hobby. And they're just fantastic.
Well, I'm
So yeah,
I'm
tell
flattered
us how
and
this
I'm-
art show came about and you're doing watercolors.
Yeah, well, it all started 12 years ago. No, that's not true. I always used to love to draw as a kid, and I think that's what drew me to no pun intended to the visual aspects of biology. I loved all the diagrams and the colors of the different tissues, etc. I collected stuff out of nature and used to observe them in little jars and stuff, just like other little kids did. Then when I got to be an adult, I reached a point in my life when I needed to fill a gap that was missing for quite a while. So I decided to take some lessons at the Art Students League, which is anybody can walk in that door, pay $56 for a month's worth of lessons from world famous artists, and without any trepidations of fear of being overly criticized because you don't know how to do anything. And I sat in Mario Cooper's class for a month without painting anything. I watched everybody else. You sit in this big square room and there might be 30 people in there and they're all busy painting and they're getting paper out and everything else. And I'm sitting there like I'd never seen any of this before. It was like, entering an operating room and you're going to do brain surgery and they expect you to do it the first time you go in there. You can't do it. And I see this kid, 6 years old, sitting on my left, and there's a woman in her late 80s sitting on my right, and they're both feverishly working away, and I've got this blank piece of paper. And Mario Cooper sits in the middle of this square, and he goes around and he looks at everybody's work, and he's got this little pencil in his hand. And if you've done something good, he gives you a little penciled star at the bottom left-hand corner of your paper. And if you've done something bad, he just keeps walking. He doesn't say anything. He just keeps going. And he says, or he'll say something like, well, I think you've done enough painting on that one. But, he'll move on. But he had these wonderful insights, which really were quite applicable to lots of other things. And he said, for instance, he asked the class, he says, what's the darkest place in the room? And everybody's looking around for the darkest place in the room. He says, well, you don't have to look too far. He says, it's actually your eyes. If you look at your eyes, not the pupils, but the irises around them, that's the darkest place. If you look into the pupil, it's even darker. So when you make your eyes, and then he went on to explain how to do eyes. At any rate, so I started out painting flowers. And I was very bad at it. I mean, I was really bad. But I must tell you, here's what kind of a teacher he was. The first time I put paint on paper, it didn't matter what it was. And I forgot. I didn't even save it. He came over to me specifically. He looked down and he put a little penciled star at the bottom left-hand corner of my paper. He says, you're not going to know if you're a painter unless you paint a mile's worth of painting. When you get to the mile, then you can look back and say, well, I didn't make much of an improvement here. I think I'll take up golf, but I kept improving, I guess. And all these iterations of the germination series started with pictures of flowers. And those are all stamens of flowers that morphed into these crazy things. And his wife, Dale Meyer, who took over his class for him after he passed away, it was a very sad event, of course. She looked down at me. I was painting flowers up to that point, and all of a sudden she looks at these things and she says, "Well, you've certainly stepped on the edge of a cliff now, haven't you?" And I just kept doing it. I'm still doing them. I love the feeling of paint on paper and the effect that it has. And I don't like messy artwork like oils. My wife loves them. They stink. You have to wash your brushes every night. You have to put your oils in the refrigerator so they don't dry out. All kinds of things. With watercolor, you can go to a river. You can take some of the water of the river. You can sit down next to the river. You can take your watercolors and you can paint a picture of the river using the river as part of the painting, which is what Winslow Homer used to do. So I just love doing this. So I use this to escape. I think this is my escapism. As you can tell, I've escaped to some bizarre places.
You still doing it? You still paint watercoloring, Dixon?
I haven't. I've done more photography recently than I have painting, but I know about. I gave you one, Vincent, and I was amazed that you actually liked it. And
no, so it's this one here.
Yeah, that's the one I gave.
The number 2, which I have a copy of it hanging in my office at Columbia. I absolutely love this. I just think the red ball, the little things coming out of it.
There's a biological underpinning to the paintings that I guess
that's going to come down here.
Can't avoid it.
That'll come to the incubator one day, and it'll be on the walls here for sure.
Kathy, thank you so much for picking me.
I am so thrilled that you shared these with us, and I think everybody needs to stop listening to TWiV right now and go look at this site, and then you can come back and listen to the rest of the pics.
So now I'm an adjunct at Fordham, and this is the art department at Fordham that actually asked the adjunct faculty, whether they had any art that they wanted to show. And so I've had actually 2 shows, and this is my second show. So I'm very proud of that. I thank all of my art teachers for that.
There was an exhibit in the lobby of our building, Hammer Building at Columbia, and you had a bunch of paintings in that as well. I remember I went and saw it a couple of years ago.
5 years in a row, in fact. That was a lot of fun.
Very cool.
Too. That was fun. Thank you.
Rich, what do you have for us?
I credit Grant McFadden with this one. It's actually a little news clip from a service called Global News that describes a new way to probe hurricanes with a machine called, I don't know if you want to call it a machine, called a sail drone. And there's a company, I think, in California that makes these called Sail Drone, which is basically a big surfboard with a big wing-type sail on it. Not all that big, actually not all that big, maybe 12 feet long or something like that. But obviously amped up with all sorts of electronic stuff that I imagine can help it navigate to where it wants to go. And they sailed this sucker into Hurricane Sam, and it took pictures and recorded all sorts of other metrics. And I'm just astonished, first of all, that this machine can survive the rigors of a hurricane. We're talking 50-foot waves and well in excess of 100-mile-an-hour winds. And so this has pictures of the sail drone, some sort of fluff with pictures of hurricanes, but then some pictures that the sail drone took itself inside the hurricane. So I thought people might be interested in that.
It's way cool. To me, the drone is a little misleading because, well, at least I think of the drone as being something aerial that's, coming down and shooting pictures. But this is sailing right in there in the water.
It's a very cool looking machine.
How it stays aright, does it ever capsize? But it must be self-righting or something.
Must be.
Yeah.
Nice. My pick is a book called A Shot to Save the World by Gregory Zuckerman. It's subtitled The Inside Story of the Life or Death Race for a COVID-19 Vaccine. And this hasn't come out yet, but I have a copy here because the author emailed me. He said, he liked TWiV. He listens to TWiV, and he said, "I'd love to hear your reaction." So this is very cool. This is about all the people in the mRNA vaccines. I think Chadox, maybe a few others. There's a list of people in the front, which is very cool. It includes a number of people, cast of characters. He's got Moderna. He's got BioNTech. He's got Pfizer. He's got Oxford with Sarah Gilbert. He has then he has a thing at the end, academic researchers. He's got Kathleen Carrico, Drew Weissman, and others. Jason McClellan is there. Government scientists. Who do you think might be on that? Tony Fauci, Barney Graham, John Mescola, Kizzmekia Corbett. Anyway, it's mostly about the people.
Oh, well, that's great.
This guy,
That
and of
sounds
course,
great.
that's the key to good science writing, right? So he went and interviewed everybody, and starts a long time ago. I mean, it starts in a parking lot in Kansas where the head of BioNTech is trying to raise money for his company years ago, you know. But you get to know these people, like the head of BioNTech and his wife. They both run the company. They're just workaholics. They sleep like 4 hours a night. That when they go on vacation, they take 5 computers with them and boxes of preprints that they read and all this. And they let their kids go swim in the pool. So it's about the people, which is interesting because you don't hear much of that. And, you know, the science is sprinkled in it. But here, the part I love is about Carrico Catalin. You know, we've already talked about the article saying, you know, she had a rough time at Penn. And this is someone in the neurosurgery department at Penn who became a rare supporter. She was absolutely brilliant, but she challenged people, and that was off-putting to people, especially those who were insecure. She was a pain in the ass. She didn't give a shit about getting a gold star from anyone. One day, Carrico was told to report to the department head. "Everyone's complaining about you," he said, calling her destructive. Carrico, who at 5'11", towered over Swain, stood up to challenge her boss. "Who's everyone? And what's their complaint? Call them now. If I made a mistake, I'd like to hear it." He wouldn't pick up the phone and call anyone. So it's that kind of stuff I love. And it's about everybody. He's got stuff on everyone. Well, they're just humans, right? And they have their own.
Yeah.
So I really like it. Love to get him on and chat about this. He's a writer for the Wall Street Journal, investigative reporter. He usually writes about business and the economy and stuff like that, investing. And he decided to do this. Love to know why. And I think it's really interesting. If you know, as David Quammen said, the best way to write about science is about the people. But this is really mostly about the people. You will hear the science and how it proceeded and so forth. But I like it. It's good.
Great.
I thought you were going to say that you had it because he had asked you to blurb it, but it's already published by the time he sent it to you. So
it's coming out October 26th. So this is a pre-publication copy,
and all the blurbs are already on it.
No, yeah, the blurbs are already on it. Oh, that reminds me. So it comes when you get these samples. It comes with a publicity sheet, right? Which it gives you bullet points that you could talk about on your show. And then they have blurbs on the back of the book. Yeah, so who's on the back here? They have Malcolm Gladwell is one of them. And then newspapers. But there's one bullet point here that I like. Zuckerman, the author, says the virus likely emerged naturally, spilling over from the animal world, and he's skeptical of the thesis that it originated in a Wuhan laboratory. I really like that. The vaccines are among humankind's most unexpected achievements and are the result of decades of dedication, creativity, and frustration. So I think he gets
Great.
it. I think he gets it.
That's good.
Yeah, there are going to be a whole bunch of these books.
Yeah.
You should check this one out. We have 2 listener picks. Catherine writes, "Dear TWiV, I was interested in Dixon's comment about the most recent IPCC report." Dixon, what's that? You're muted.
Oh, you're muted.
Oh, Intergovernmental Panel on Climate Change. Okay. in the time. You're still not unmuted, but I was able to Google it in the time. I'd like to recommend Project Drawdown, and gives a link, plus the book, which is a great resource. It ranks various climate solutions by impact to help understand the most important things to do to help stop climate change. It makes you feel better to work on solutions. Okay, Dixon? So check that out. Project Drawdown.
Thank you.
Sincerely, Catherine, just an energy efficiency engineer.
Huh.
Charles writes, Ave TWiVumvirs, those who are about to plaque salute you. That's cool. Or TCID 50, right? I must confess that I am quite far behind on TWiV. Aside from the few I've attended in person at ASV meetings, I only started listening in earnest a few years ago. Currently, a vast gulf of time separates us as I work my way through your extensive archive. Your broadcast started around the same time that my adult career as a virologist began, as a graduate student at Colorado State University. It's been fascinating to follow the virological highlights of the past 10 years as my career has progressed along with your program. Thank you for documenting the weather, including your experience during Hurricane Sandy, which welcomed me to my postdoc in the laboratory of Eckhard Wimmer. Not sure if this has come up, but for my listener pick of the week, I would like to suggest Aerosmith by Sinclair Lewis.
Oh, yeah.
As phage therapy comes back into the limelight, it's important to recognize that this idea is over a century old and even reached public consciousness in this early 20th century novel about a microbiology graduate student and his irascible German professor. I would also recommend another book from the 1920s called Microbe Hunters, written by Paul de Kreef. These books were highly influential during my childhood and influenced my decision to become a virologist. This is Charles Brandon Stauft. Yeah, I love those books. Those are all great.
Yeah, absolutely.
Yeah, so Aerosmith has a connection to the University of Michigan because Paul de Kreef, who is the author of the other book, Microbe Hunters, was a student with Frederick Novi, who founded our department. And Vincent, I don't know if you remember hearing about this in the lecture.
I do.
For the milestones of microbiology that you came for.
Yeah.
But de Kreef collaborated with Sinclair Lewis on Aerosmith, and he gave him not only some of the scientific background about phage and bacteria and so forth, but also the important insight into the sociology of the scientific environment. So that's our connection to it here at Michigan.
I remember that. Yeah, I remember that story during the milestone ceremony. Yeah, that was cool. Yeah, I have my mother was an English teacher in high school, and she used to teach this book, and I have her copy all marked up in pencil with things underlined and things she wanted to highlight. It's really a treasure. It's great. So someone this week talking about, following things with TWiV, someone this week said, the TWiVs of the past year and a half are a great record of the pandemic in a way, because they're every week and we talk about it and people could go back and they said you should archive it somewhere because the copies in my basement won't last forever. It's true. It is an archive of the pandemic, right? We've been pretty much documenting it in ways.
Yeah, I was reflecting this morning on all of the ideas that we have had along the way that have proven wrong. So if you go back to the beginning and listen through and see the evolution, you see, that we were cluelesser then than we are now, and it's been a tortuous journey.
But it is a record that really you can't find anywhere else.
Yeah,
right?
yeah.
2-3 times a week by virologists chatting for 2 hours.
Yeah.
That's right.
Some may say it's a record they don't want because it's too much, but no, you don't get to choose that. All right. That is TWiV 815, microbe.tv/TWiV. You can send your questions and comments to TWiV@microbe.tv. And if you like what we do, consider supporting us, microbe.tv/contribute. Dixon Despommier, Racaniello.org, Despommier.photoart.com. That's one I haven't given in a while.
It's true.
Thank you, Dixon.
Thank you, Vincent. And you should mention the fact that without you, I wouldn't have those websites. So thank you so much for your collaboration.
My pleasure, Dixon. And we have something else to do, I think you asked me. I forgot.
Yep.
Okay, no problem.
Right.
Kathy
Good.
Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Fair enough. Always a good time.
Good, Rich.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.